Hello, welcome to another bioinformatics video. In this one, I'll be going through the exercises related to phylogeny and evolution. And the first function simply asks us to, the first question simply asks us to write a function that computes the Hamming distance between two sequences. And the Hamming distance is simply the number of substitutions needed to make one sequence into the other, or the number of mismatches, basically. And one requirement is that the two sequences have to have equal lengths, because this function will not take into account deletions or substitutions. It will simply compare the two sequences position by position and check for match or mismatch and count any differences between the two. So this is how you can do it. Real simple. So we have a function here. It takes two inputs, sequence one, sequence two. We have a result variable here, which is simply the count that we're going to get, the number of differences. So this is our Hamming distance, but initialized at 0 to begin with. And then we have a for loop that will go through a range from 0 up until the length of either one of the sequences, since they're the same length. And for each position, it will check the base stored in that position in sequence 1. And it will likewise check the base stored in the same position in sequence 2 and ask, are they not equal in this case? You could have done this also, check if they're equal and then have an else case, but this is slightly more condensed. So if we have an exclamation mark here plus an equal sign, this means not equal. But we could have easily just have done this. Just like that. That would also have worked equally well. So however you did it is fine. And then down here, the result is incremented by 1. In any case, a difference is detected, and then the result is returned. So real simple. And here we have two sequences, and we're told these are from the nucleocapsid phosphoprotein of SARS-CoV-2, and they're taken from different time points. So one of the sequences is from real early on in the pandemic in 2020, and sequence 2 is from the year 2021. So a more modern sequence. And we can see that for this particular protein, there are 3 mutations difference, 3 substitutions. So they haven't changed much, but in the case of this protein, they have changed a little bit. And problem 4.2, make a distance matrix from the Cas9 sequences that we explored in the previous exercise. So we have this big list of Cas9 related sequences. And we want a distance matrix that that we can print out and then produce a tree from. So a distance matrix is simply a matrix where every row and every column correspond to the 2 different sequences. What do I mean by that? So if we look at this matrix here, the rows here are going to correspond to the different sequences here, and then the columns are going to correspond to those same sequences. So we have everything compared with everything, and then each element, each square of this matrix is going to be either a Hamming or Levenshtein distance. And the Levenshtein distance, which you don't need to, if you're in the course, you don't need to know how we made it. Simply know that it gives you a distance metric that accounts for insertions and deletions. So it is just like Hamming distance in the sense that it's looking for substitutions, just the number of differences between 2 sequences. But a Hamming distance will be really susceptible to frame shifts. So if we were to delete, say, one sequence near the beginning of the sequence, if we were to delete one base near the beginning of the sequence, then the Hamming distance would take that frame shift and just see mismatches all the way throughout the frame shift. The Levenstein distance is smarter and kind of similar to some of the pairwise alignment algorithms we've been working with. It will understand that that an insertion is needed in such a case. So it will compute the number of changes you would need to make the two sequences the same, including insertions, deletions, but also substitution, the number of edits. So this is why it's sometimes also called the edit distance. So each element in this matrix then is a Levenstein distance between, in this case, say sequence 6, and if we're looking at the square, and sequence 10. And then instead of sequence 0, 1, 2, 3, 4, 0, 1, 2, 3, 4, it's going to be these guys here, Cas9, 513, 516, and so on. And so we'll be checking how many edits you need to make between each sequence to make them into another of these sequences. So how do we do this? We can make an empty matrix. So the numpy.zeros function we can use to just supply the dimensions and create an empty matrix. Take note of the double parentheses here, which can be kind of trip people up. That is because if we want to make a two-dimensional matrix, NumPy requires us to supply the dimensions as a tuple or a pair of numbers in parentheses like this. If we only supply one number, it understands from that that we want a one-dimensional matrix. But if we supply two numbers without the parentheses, then it will get confused because it will see this first one and think, oh, we want a one-dimensional sequence, and it will see the second parameter and think that this is some additional argument, such as the data type, which you can also supply to the zeros function. A bit of a tangent there, but anyway, we make this empty zeros matrix, and we're going to use, as usual with 2D matrices, to navigate through them, we will use 2 for loops, and each is a square matrix, so we can just have each one loop through a range from 0 to the length of the matrix. And so it doesn't matter if the length of the matrix is the number of rows or the number of columns, they're the same, so we just use this. And inside the loop, we're going to be changing the values at row I, column j in the matrix. So whichever I and whichever j we're on, whichever row and column we're on, we will go and look that position up in the matrix and make a change. And so initially they're all zeros, but then after we go through it, we'll be inserting the Levenstein distance in each case. And the Levenstein distance function works just like the Hamming distance function that we made. It takes two sequences as its input and produces a single number or distance as its output. And in this case, the sequences we want are going to be coming from this list Cas9 vals here, which is simply the sequences without the IDs, and fetching whichever row we're interested in and whichever column we're interested in, fetching the corresponding sequences from this list, and then comparing them and assigning that to the square in the matrix. So if we run this, it takes some time because Levenstein distance is a much more involved calculation than the Hamming distance. But at some point it finishes, and then we get to run this block that is just given to us. And all this is going to do is display the matrix with our choice of color scheme here, the spectral color map, and also print it out. And we can see, for example, zeros along the diagonal. Why is this the case? Well, the edit distance between a sequence and itself is always going to be 0, right? It takes 0 changes to make a sequence into itself. So anytime you're looking at, say, sequence 4 compared with sequence 4 here, the edit distance is always going to be 0 in that case. And then finally, we run this last line here. And again, we don't need to think so much about the details, but what this will do is it will run a hierarchical clustering algorithm from the SciPy library, scipy.cluster.hierarchy. We will run this linkage function, which will take as its input a distance matrix, and it will do all the work of producing this linkage diagram or hierarchy or tree, whatever you want to call it. And we do it twice here. So we display the linkage diagram with the labels. So we can fetch the Cas9 keys from up here that are simply the list of IDs. And we can do another one where we don't supply anything to the label list, and it will just apply the numbers, so the order. So both can be kind of useful for understanding what's going on here. But these are the exact same phylogenetic tree or distance tree. So yeah, that's how this problem is. And we can see which sequences are more related to each other, at least in terms of sequence similarity or Levenshtein distance. Finally, problem 4.3. In this one, we have a Pssm, or a position-specific substitution matrix, also called position-specific score matrix. And if you remember, these are a matrix where you have the rows are all the amino acids, and the columns are positions in a protein. So we have a 16 residue long peptide that we're considering, and the individual elements in this matrix are the substitution scores, and those scores are log odds probabilities. So the log of the odds of making a particular substitution, for instance, for an A here, the ratio of that probability with whatever the base mutation probability is. And those odds are then presented as a log to generate these scores. And so the matrix, the score matrix here, it's all these substitution scores, and we can see they're changing. So they're not the same for every position. So unlike, say, Blossom 62, which is just a uniform substitution matrix that does not take into account any sort of context, in a Pssm, we have a particular kind of customized substitution system for every single position. And the point of this problem is to start to think of this as from a kind of evolutionary perspective. So the fact that the probabilities are not equal for the different amino acids, for example, the F here in the first column is really strong. It has a high value. That means there's a strong evolutionary preference for this particular amino acid type in this position. And so we can start to think of those as evolutionary constraints. So mutations may occur or try to occur, but they're going to be influenced by the different rates and the different positions. So this problem, the material under the hood was a bit complex. So the point of this problem was more to be just playing a bit with the parameters. But basically what we've set up here is a small simulation where we have a 16 base long peptide. We have a mutation rate here, average mutations per generation. And we can pick a number of generations here, for example, 40,000. And then we have a Pssm. our matrix. So these are our 4 main parameters. And then what this will do is it will go in and things mainly start on this loop here. It will go in and for each generation simulate the evolutionary process as a stochastic process. So we'll be simulating random mutations using the random library here from Python. So for each generation, we have a list of lineage sequences starting with our first one. And what we're going to be doing is keeping track of every generation. So if a change happens this generation and we replace a residue with a Q, for instance, we'll have a record that is, we had this first guy, and then the second generation, it was the one with the Q in it instead. So we'll be just keeping a record in this lineage seeks initially empty list here, and it will be adding to this something that we've called current gen seek. So that is the current sequence of the most recent generation that's going to be changing as we go through this loop. And then for each one of these generations, we're then going to go through every single position. So the columns in these, or the positions in the original 16 base long peptide, we'll be going through each of the positions and generating a single random number, and then asking, is it less than the average number of mutations per generation, our mutation rate, basically, that's up here. And then checking the probability that a particular substitution will occur. So if, say, so like how this could work is if, say, this random number is less than the mutation probability here, then we decide that we will go ahead and create a mutation. And so the effect this has is that this random process will happen as often as the mutation probability demands. So if we, it's 0.01, about 1%. So if we did this, 1,000 times, we should expect approximately 10 of these times to trigger this, this condition here. So this is how we can simulate a random process. And then if that step is triggered, so then we've decided that a mutation will occur at a particular position, then the question goes to which substitution will we take? And then for that, we go into our Pssm and determine what the relative likelihoods of each amino acid are. And of course, if it's a substitution, then we'll be omitting the identity of the current amino acid that's there. So that's what this line is doing. It's just checking which amino acid to set to 0 probability so that we don't sample the same amino acid, because we've already decided that there will be a substitution. So given that condition, we should decide on a different one. And so then we'll run this random.choices thing that we used in an earlier exercise, if you recall, to locate where the EcoR1 cut site would be in a random 100 base long protein. We can run the exact same function here. In this case, in order to choose a member of this column. And in this case, it's very important to utilize the scores to influence that probability. So here we've converted the log scores into actual probabilities by up here raising everything, taking 10 to the power, the score that's there, and then normalizing it by dividing by the sum. Yeah, so getting a bit detailed here, so I don't want to go too into detail, but basically this will pick a base and then propose that change. So we'll implement the change and update the current gen sec here, current gen seq. So the sequence here will then be changing. And if we look through, if we kind of scroll through the results, we can see that the first generation, for instance, has an L here, and then the second generation has a Q there. So substitutions are progressively taking place, and the sequences are getting more and more different from their original ancestor. And then finally, we have this timeline array here, and all that's happening here is it's initially declared as a zeros array. So it's just an empty array with as many slots as there are generations. So if we do 40,000 generations, timeline is a 40,000 long list of zeros. And what we'll be doing is updating the values in timeline as we go through the loop with Levenshtein distances from whatever generation we're on to the original sequence, this one here. And so this gives us a kind of time series, not time, I guess, generation-based diagram of how different the sequence is relative to its ancestor. And if we run it for, just 40, we can see that there's quite a bit of change in the beginning. If we run it for longer times, we begin to approach, we begin to see what the kind of equilibrium state is. So eventually it kind of stops changing much, and we can see that it's like fluctuating in place here. And the last thing was to change the Pssm. So we started off with this Pssm, and then we're asked to change it to a different one. And we get a different behavior, seeing that the kind of evolutionary pressure is very different for this one. So when we, for this particular sequence, when we kind of change things up, so now, this F is no longer as favored as it was, for example, here. So we have less pressure, for example, for the first base to be the F relative to the other Pssm. And it's more different from the original sequence, so we see greater divergence. So that was the idea behind the problem. And we could have just visually confirmed, so it's about an average of 12, Levenshtein distance of 12 if we look here. But we could also take an average of this timeline variable to measure the actual, the average Levenshtein distance. And being careful not to get the beginning, right, because it's changing a lot in the beginning. So if we measure from, say, if we do 40,000 iterations and we measure everything from time point generation 5,000 onward, then we should get a pretty good estimate of the average distance relative to our original. And okay, and then we can see that the average here is 12.1. And visually, you can also see it's pretty much centered around 12 here. So that's the third problem. And then the challenge problem, the goal was to go to NCBI or NextStrain and to fetch sequences related to coronavirus from different time points and construct a tree. And so this problem is a bit open-ended. So I will just show how to do this through NCBI. So if we Google NCBI and just go to their main page, we can see that they have currently, at least as of March 2021, they have on their website a link to SARS-CoV-2 data. And if we go down here, we can see that there's a section here for nucleotide records. And here we get this nice browser. So each of these entries then is a particular, zoom out, each one of these entries is a particular sequence, a different individual experiment or sample. And what we can do is apply a few filters here, I would say the most important one is this nucleotide completeness. So in order to not get partial records, we'll check this box and then filter results so that we get the most complete ones only. And then I will go down here and sort by collection date. Because I think this is maybe the most interesting variable. Some of the sequencing, I think there is the sequencing or published dates are probably also kind of similar to collection date, but I think collection date is probably the most important variable if we want to capture different points in time during the pandemic. And then what we can do is, so we have 276 records or pages of records here. Let's just pick 5 of these. And I've already done this in my example, but I'll show how we do one of them. So we just pick one of these records here. And this gives us a whole coronavirus genome. So we have, for example, translated proteins for all the proteins, open reading frame 1 protein, the surface glycoprotein as well. And then at the bottom, the full genome as a nucleotide sequence. And what we can do is just pick a protein of interest. So I chose here the surface glycoprotein. That's the spike protein, I think. Separate, yeah, I believe that is the spike protein. So if we just search for surface again, and what I want actually is the DNA. So we could use this protein sequence here, but you know, there are not so many changes, right? We're dealing with very small evolutionary distances, and so we're kind of thinking on the scale of, you know, 1 or 2 point mutations for one of these proteins. So we might not see much if we look just at the protein, because you know, maybe some silent mutations will take place that do not change the codon, that do not change the amino acid, even though the DNA is different. So to capture more variation and produce a more interesting tree, I will take the indices here for the gene. So this is the start and end bases for this surface glycoprotein here. So I'll just take these along with the accession number, the GenBank ID that's up here. So those 2 pieces of information, and then I would just do this for you know, 5 random sequences from different time points. So just trying to spread things out. Maybe I would go to, you know, page 75 or something like that and fetch 1 record from there to gather a kind of representative bunch. So that is how I did this, and I just made this list here of IDs. So these are, here we have the GenBank IDs, and just copying the same kind of BioPython entrées code here that we used in previous exercises to fetch records and sequences. So just kind of reusing that code. And then here I have what I'm calling the gene list. And this is simply a list of lists. And each sublist is a pair of indices. And so we could use this to fetch, if we can get the sequence, the whole genome sequence from one of these records, we could fetch the individual gene then by supplying these indices. And so I have a loop here that's just going through our ID list, then going to entres, and running this efetch line here. And take note of the correct database. So if you're looking for protein sequences, this should be the protein database. And you can check which database you're looking at by scrolling all the way down to the bottom on NCBI. And here you can see that we're in the nucleotide database. So fetching these records from the nucleotide database in my case, and then supplying the correct ID from the ID list. So using the index here for my range for loop to get the different positions. And then I create here this gene start variable just to make things clear, which is going to be the first number from these pairs of the gene indices. And actually in this one, in order to use the Hamming distance, I would like to use Hamming distance. So we could use either Levenshtein or Hamming distance. But if we wanted to use Hamming distance, for instance, all the sequences need to be the same length. So rather than actually supplying the full indices from start to finish, what I've done instead is just provided the start of the sequence, the start index, and then added a fixed plus 4,000. So if you see most of these, it's about 4,000 bases long. And then I close down this handle for each item. And so the loop will go through and do this for all of them and produce this list at the end, SARS-CoV-2 sequences. And these will all be 4,000 base long sequences, approximately encompassing the surface glycoprotein. And then finally, I make this distance matrix here, just as we did in the previous, in problem number 2. It runs really fast, right, because I'm using Hamming distance in this case. It's a simpler calculation, and we get our distance matrix, and we can see here it's quite diverse. So that's a good thing for our hierarchy. And then I run here the clustering, the hierarchical clustering, and we can see a nice division of differences. Yeah, so that is one way to do this. So you could have done this also looking at the proteins, and this looking at the proteins might be actually a more interesting way to perhaps identify strains of interest, like phenotypic or functional differences. So for instance, we have these new strains that people are worried about that are behaving differently and yeah, reacting differently to antibody treatments. So that information might not be as obvious in the DNA sequence because the DNA is always changing, but not necessarily with consequences. If we had looked at the proteins, perhaps maybe we would have seen something more meaningful, but probably less change as well. So if you tried with the proteins and just saw everything looks the same, that would not surprise me either. So that's it for this video. Thank you and see you in the next one.